From Microbe TV, this is Office Hours, and today is Wednesday, February 28th, 2024. Oops, Nels is not with me. Let me erase Nels there. I'm Vincent Racaniello. I'm not a Nels Vincent recombinant. I'm Vincent Racaniello, and welcome to your corner of the viral internet where you can get virology questions answered. You can get speculations on almost anything else because I know viruses. And speaking of virology, I think I mentioned this on some live stream. I was having some blood drawn, and the nurse said, "Oh, I see here that you're a virologist." She said, "What's that?" My jaw dropped. Didn't everyone know what a virologist is, especially after pandemic? And she didn't know what I did. So that made me want to do these live streams and my podcast even more. Anyway, we have a night of interest ahead of us as usual, which is not just me saying things and answering your questions, but you chatting amongst yourselves. So let me welcome our moderators tonight. We have Les, we have Steph. So Les is from California. Steph SF, San Francisco, of course. We have Vanity Nutrition from the New York City area. Who else do we have? I can tell by the blue. Tom Steinberg. Not a recombinant nor a chimera. Nope. I like recombinant, right? Because it draws from my viral background. You know, kids are recombinants of their parents, right? Get genes from their mother and father. So there's shuffling of the chromosomes, but sometimes also some recombination occurring. You know, a chimera is usually like a human-animal hybrid, which, you know, like a guy's head and chest and arms stuck on a horse's body. Crazy stuff. And so someone wants to know how the moderators work. That's Jessica. I'm going to ask where everyone's from, but how do moderators work? Well, they're here, and they have their blue names are in blue, and they have a wrench next to their name because they're going to fix things when they break. And what is break? Well, if someone is very obnoxious and says, like, Vincent, you don't know anything about viruses, they're going to be timed out, and if they keep it up, we'll block them entirely. And if people say filthy things, filth is just not the right word, but you know what I mean. Sexually oriented things, inappropriate things. And sometimes bots come in and spew pornography here in words, not pictures, words. So they cut them out. So the mods are great because we have a pretty busy stream here. Hey, when it was 1,000 people, oh, they had their work cut out for them. So they're very good, and they would like to also listen and participate, and it's hard to do both. So I appreciate their service, for which they are not paid. And I greatly appreciate that. But where are you from, folks? Let's see. I have a few things to tell you tonight, but I want to find out where people are from. And, you know, so you guys have a pre-stream chat going on here, right? And sometimes those questions don't make it to when I start the stream. A lot of them get cut off. I don't know what the algorithm is. I don't know who decides what, not who, I don't care who, but how it's decided what comes through or not. So if you put a question in earlier and you don't see it, just ask it again. I don't have a problem with that. All right, so where are you from? I know that Carol is our favorite nephrologist. She's visiting her mom in Sacramento, but Carol's from Southern California. There you go. Where it's sunny and 65 degrees F. Actually, here it got warm today. It is not sunny anymore because it's 8 p.m. The sun has set. It's 13 degrees and windy. It's been raining on and off all day. I spent my time running around the city today. This morning I went up to Columbia University Medical Center for a bunch of meetings, and then I went down to teach my course, and now I'm back here today. Tom is in Madison, Wisconsin. A lot going on, may not be able to stay, but thanks for your time anyway. Yeah. Who else? Lise is from very windy Columbus. Hmm. Laura is from San Francisco. She visited the incubator earlier this month with her daughter, who lives here in New York. It was great to see you, and everyone's welcome to visit the incubator. This is a cool space, as you can see from the cool lights and artifacts behind me. Um, all stuff from my career, but there's more to it. There's a cool studio here, a recording studio with lots of equipment that all of you have contributed towards. It's your work that helped us to build this incubator. And then next door we have an office, a common room with a big Ebola painting. We have a kitchen and a library. It's cool. And I talk a lot, so you'll be hearing stories from me because I just spontaneously spew stories. Yeah. So good to see you. Abdulaziz is from Saudi Arabia. Welcome. Dagfinn Rosnes is from Oslo, Norway. 2 degrees C and snowing. Wow, wow, wow. Snow. Now that it's not snowing anymore, I miss it already. Will is from Changsha, China, where I hope to visit someday again. Not again, for the first time. I canceled my trip last time. That would be fun. Here's Jonathan from Encinitas, California. People are still talking about bubble tea. We talked about that last week. Sorry, that's Andrew, our moderator. I forgot to mention Andrew. I didn't see him. Thank you, Andrew, for moderating tonight. Frank is from Santa Barbara. Simon's in the Bay Area. Leo is in Mendocino. We're going to talk about Questella's whack flu. She calls it whack flu. We're going to see what the heck that's all about. Today is the 28th. Tomorrow is Leap Day, February Leap Day. Neva is from Buda, Texas, where we're going to be in a little over a week. I'll tell you more about that. Very cool. Gang is from Chicago. MK is from Eastern Massachusetts. Nicola is from Italy. Wacky weather everywhere, right? John is from Minneapolis. We're at 16F. Joseph is from Ontario. Karen is in northern Maine where it's stupid warm, 37F. Wow, the dog race is canceled. That's too bad. Nebby is from Florida. Artemis is from Montreal, and we did meet in person. Ronald is from Galveston, Texas. You have to be kidding me that a nurse did not know what a... Yeah, she was a nurse. She had RN on her ID. Not too old either. I mean, I would say she's in her 40s probably. So the school is not far behind her. I don't know. I just don't get it. Peter is from Boulder, Colorado. Airy Ozone is from Asheville, North Carolina, where I haven't seen any centaurs. I like when people say things like that. It tells me you're listening to me when people make little comments, right? There are no centaurs. That's great. Typically, a phlebotomy tech draws blood, not a nurse. No, she was not a phlebotomy tech. I didn't really tell you the whole story, but she was a nurse who was, before the doc comes in, the nurse talks to you a little bit, goes over your chart and stuff like that. But she had RN on her ID. I mean, yeah, okay. Hey, wait, there are legit biological chimeras. Yeah, I know, but I'm not a chimera with nails. Even if I were, it would be a recombinant. Rip de Campus from Taos, New Mexico. Cool place. Hi from the UK from Claire. Hello, Amy. Amy is in the D.C. area, let's say. Laura is from Chesapeake, Virginia. Tona is from Long Island. Cheryl is from Santa Rosa. And Leo says we love Amy. Luis is from Portugal. Ooh, Giza Brakpal is from Phayat, Thailand. Wow. someone from Thailand last time, too. Welcome. Must be like morning-ish there, right? Cognition Today is from New Orleans. Luis is from Portugal. Yeah, we already got Luis. Adam is in Canada. Hat is in Los Angeles. Ziziva 303 is from Denver. We didn't get Ziziva there. It didn't click. Elizabeth is from West Virginia. Jessica is from Toledo, Ohio. Jacques is from São Paulo, Brazil. Welcome. Jan is from Tucson. Hans is from Ecuador. Lauren is from Edmonton, Alberta. Alfred is from Hong Kong. Wow, cool. Did Luis get... Yeah, Luis has told us now 3 times where he's from. I got you, Luis. I got you. Mike and Lori Ding are from Seattle. Lori and Mike. Oh, John says, even though it's no longer Q&A with A and V, it's comforting to know Amy is here. I just wish I was smart enough to qualify for her lab assistant job. If you guys have a question I can't answer, I often will text it to Amy, and then she will text me back and make me look smart. Yeah. Thank you, Amy. Sean is from Tennessee. And Rip the Comp says, shout out to Lafayette, Colorado from Taos. Patricia is in Massachusetts. Calliope. Hello, Calliope. A goddess of what I forgot, but a goddess as well as an instrument. She was on the Tweevo stream, yesterday that was. Wow. From Toronto. Robin is in Norwich. Helmug. Lori Pueblo, Colorado. So this is Lori from Pueblo, Colorado. Give us a virtual tour one day of the incubator. Yeah, I could do that. I'd need to get a portable camera I can walk around with. I could do it with my phone, so I could actually transmit from my phone to this stream and walk you around. That would be cool. I got to figure that out ahead of time, but that would be cool. Marianne is from Mableton, Georgia. And I know Rach is from the Isle of Man, but we'll get to your question later. Lauren is from Edmonton. Wei Jing is from Oakland, California. John is from Southampton, Ontario. We'll get to Costella's whack flu. Just hang in there. Environmental Coffee House is from Angelica, New York. Paul, Patty. Hi from Patty in Massachusetts. So PGL is Patty. There you go. Yes, yes. What else do we have here? Razor is from Saskatchewan, Canada. Callie is from New York City. You should come to the incubator and no, then you couldn't chat. Well, you could be next door and have the feel of the incubator and look at the stream on your laptop. That would be fine. That would be fun. Let's see. That's Kelly. Kelly Wong. Very cool. Be careful disparaging nurses. I'm not disparaging them, but I'm just saying they should know what a virologist is. Okay? Is that too hard to ask? Especially as a virologist? Rafael is from Brazil. Another Brazil. Excellent. Excellent. I'm not disparaging nurses. I don't disparage anyone, Steph. You should know that. I'm a non-disparager. All right. I know some people will disagree with that. I disparage when people say things that are not right. Oh, boy, I went on rants today in my virology lecture. And that guy raised his hand. I said, wait, let me finish my rant. Troy is from British Columbia. SRR is from Paso Robles, who's on a date night. It's a TWIV couple. Hey, cool. That's just great. Date night. Lovely, lovely, lovely. I love it. You remembered the best part, the goddess. Of course. How could I forget a name like Calliope? And then you add Nikitas. I have a Greek woman in my class, and I can't remember her name. When I see her, I say, "Oh, you're the one with the Greek name." Uh, Kyriaki is her last name, I think. No, maybe Kyriaki is her first name. Yeah, maybe. Anyway, um, Monica is from Colombia, South America. We've had a couple of Colombians here lately. Eliza's in Toronto as well. Peter's going to ask for the same incubator tour, the virtual one. All right. Let me write it down on a piece of paper. Maybe next week. I don't think it's so hard to do that. Virtual incubator tour. So, this live stream is almost like my class, except there's no noise. There's always a little noise in the class. People ask questions. I hear voices. I'm here myself, and it's very quiet because this is soundproof. It's night here. There's nobody in the building anymore. It's different, but it's fine. I'm not complaining. Not complaining at all. Tona says, in her defense, she probably knows what virology is. She just doesn't know in practice what a virologist does. Maybe she just asked me, what is that? That's all. And I kind of repeat it. It's like, there are plenty of professions that I wouldn't know of, right? Why don't you guys find one that's really a strange name and I'm not going to know it, right? Like, what's a philologist? Let's see. Philologist. Someone who studies the history of languages. Okay? And what do you call someone who studies stamps? Philatelist. I don't know if that philatelist. Philatelist. I'm reading this phonetic. I don't know the philologist. I don't know what that is unless I look it up. So I'm sure there are others as well like that. Okay. Martha is signing on from Charleston. EHS is in Lehigh Valley, Pennsylvania. Philatelist, and then philatelist. Yeah. Yeah. Okay. Philatelist. Well, what are we talking about? Stamps. Yeah, philatelist. I looked it up because I didn't know. What was the other one? The one, philologist. Yeah, the philologist is the one who studies language. I wouldn't know that. I hadn't heard philologist. So there are lots of professions I wouldn't know either. But having just gone through a pandemic, wouldn't you have seen a news program somewhere saying, Dr. So-and-so, virologist at Mount Sinai, says, right? Okay. Horologist. What's a horologist? I only know a horticulturist. This is what I like about the live stream. I can set you guys off on little side things. It's great. We will get to viruses for sure. A strange profession here is bryology. What is that? The study of bryophytes? Yeah, bryophytes. Bryologists are people who study bryophytes. That's not a useful definition, is it? Well, I said to the nurse, a virologist studies viruses. So, bryophytes. Do you know what bryophytes are? What is a bryophyte? It's a kind of a plant, a moss, a liverwort. Mosses, liverworts, and hornworts. All right, well, this is not This Week in Plant. An oenologist, yeah. All right, what else do we have here? An entomologist versus etymologist. So an entomologist, of course, studies insects, but what is an etymologist, studying the structure of words or languages? It looks like it would be similar to what did I say? Philologist? I can't even remember. Philologist. Calliope. Okay, Calliope comes to the rescue again. Philo, like, lover of logos is word, lover of words. Thank you. Oh, numismatist. What is that? What is a is that a stamp person too? Numismatics. Wait, it's there. Numismatics. I can't get it. I can't get it. Why do you think I'm such a good live streamer? Why? Tell me why. Because I yak with you guys. I come up with stuff and we talk about it. I'm just curious. That's all. You can't be afraid to talk. I can just come in and sit here with nothing planned. And I do have things planned, by the way. But I can just come in and start talking because I got a lot spinning around up here. It's fun. I just love it. An oenologist studies winemaking, right? Probably the nurse wasn't listening at all and asks everyone. No, she had it on the chart, right? It was written on the chart that I'm a virologist. Cool. All right. After working in hospitals for so many years, the number one disparager of nurses is other nurses. I have to say, I got a nurse angry on by email once. She wrote in and she complained about the sound on one of the episodes. And I guess I was in a bad mood. I said, I understand that it wasn't great. I have a lot of stuff to do, and sometimes I screw up. And she got insulted. And then, just like that was a year ago, and then just a week ago, she said, "You see, I don't know. There's something recently. Nurses got disparaged somewhere." And she said, think about this the next time you disparage a nurse. I wasn't disparaging her as a nurse, just as saying that my sound was lousy. But I feel very guilty when people tell me I'm disparaging them, right? Hey, this is great. People who can't distinguish between etymology and entomology bug me in ways I cannot put into words. Oh my God, that's just great. Calliope must be an etymologist, right? Because she says anything that starts with philo, just think of hydrophilic, likes water. That way you will know a bit about that word. She means, likes, like liking stamps or something. So, Calliope, are you some kind of a philo? Word origin. Lori is from Tomball, Texas. Artemis ethology, the study of behavior of animals. That's a new one. I didn't know that. Vincent is a podcastologist. I don't think that's a word. I don't study podcasts. I do them, right? But it's okay. Costello says, you let people talk and don't get mean or a big head and everything flows naturally. Thank you. I appreciate that. Those are that's the way I like to do things, for sure. And Kip says, hi, Vincent. Both you and I enjoy horology. I don't know what that is. Horology. Science of measuring time and the art of making timepieces. Yeah, I do like watches. Yeah. Okay, so this one was made in Germany. Can anybody guess what it is? It's classic, made in Germany. A numismatist, I don't know how to pronounce it, studies coins. Okay. All right. All that stuff spinning around up there is why I'm here. This is what I love. I said I had a lot of stuff spinning around up here, and then Troy picks up on it. So you like to hear it. I mean, I appreciate that you guys like to hear this. What do we have? 164. That's a lot of people that want to listen, more than my class. My class isn't that big. My class is 110. So I like that. Melitologist, the study of bees. Yeah, melatonin is a substance from bees. I know that. Sometimes you can make light of things. Yeah, herpetologist studies reptiles, right? Calliope is Greek. And all these words have origins from Greek. A huge advantage knowing Greek and or Latin. Yeah, so poliomyelitis has both Greek and Latin roots. Polio. So myelon is gray. I think that's the Greek. Now I've forgotten what polio. Of course, is inflammation. I think that's a Latin root. And the polio. I forgot what good is it good of me to forget what the polio part? Amy, go look it up on my lecture where I define poliomyelitis and tell you what all the Latin and Greek roots are. But Calliope, myelon sounds like myelon sounds like a Greek root, doesn't it? Okay. Richard stole that joke from the internet. It's really good. I really like it. and you had good timing with that, right? It was perfect fit for us because we had mentioned the two etymologists, and well, we didn't mention entomologists at all. This is good. Hi, MN. You're that doesn't matter. You're never late to this live stream. Let's see. Anyone else want to tell me? I bet Rianne would ace this category of weird professions. Yeah, that's a good one. $500. I don't know how you do that Jeopardy stuff. We did it at TWIV 1000. Yeah, the Greeks have a word for everything. I think it's great to know Greek and Latin because, as Calliope said, you know lots of roots. All right, so let me tell you some news first. We're going to be going to Texas in not this weekend, but the weekend after, and doing a couple pods. So let's put up the pods here. So the first one is TWIVO 100. That's going to be Sunday, March 10th, in Johnson City, Texas, at the Science Mill, which is owned by Nels' parents. It's a science museum for kids, and we will. The museum closes at 4, and we will do a TWIVO there with a guest. And you're welcome to come. We can take up to 30 people, and there'll be a reception with food afterwards. You can RSVP at microbe.tv. Yeah, so the email is rsvp@microbe.tv. And that's Sunday, March 10th, about 5 p.m. Okay, so that's TWIVO. So if you're in the area, please come, but RSVP, because we'd like to get an idea of how much food to order afterwards. It'll be fun, right? We're going to have a guest. I'm not sure who it is yet. Nels hasn't told me. We'll have some people from the museum to explain what they're doing and, the usual banter. So it'll be fun. So that's Sunday. And then Monday, TWIV, it will be at the University of Texas. And Rich will join me. I think Rich is also coming to the TWIVO, by the way, because he's driving us from Austin. So I think he should be on the TWIVO. So Rich Condit and I will speak with Jason McClellan, who, you folks should know who he is. But if you don't, I understand. He's a structural biologist at the University of Texas, Austin. He's been on TWIV 3 times previously. And, he made a discovery that was very important for the production of mRNA vaccines against the SARS-CoV-2 spike. He had previously found that for another spike protein, putting 2 prolines in would help stabilize the protein in a pre-fusion conformation. And what does that mean? Well, when the spike binds its receptor, it undergoes a conformational chain that primes it for fusion. So the virus in the cell membrane can fuse and the genome can come out into the cell. And you don't want the spike protein doing that. You want it to be in its native state where the epitopes, the protein parts, are exposed that are going to be generating antibodies. So putting 2 prolines in makes it stay in the prefusion state, and you get much higher antibody levels to the neutralizing epitope. So, but we'll talk about other things most likely. All right, so that's Monday, March 11th, and it's at 11 a.m., and it's in the Norman Hackerman building. There's a big seminar room when you go in there. You'll see it. You don't need to RSVP about that because that room can hold many people, and we did TWIV 500 there, actually. All right, so those are the 2. And then Tuesday, is it Tuesday? Yes, I will do a session at South by Southwest, actually. So that's not a microbe TV thing. Let me turn this off so you can see me. That's not a microbe TV thing. I was invited to do that. And so, South by Southwest is a media conference, right? It's big. A lot of people flow into Austin. And they want to do more science. So they want me. I'm going to have two guests, and we'll do a conversation in front of an audience. One of them is Stephanie Strathdy, who you may know. that wrote the book The Perfect Predator, and her husband his life was saved by phage therapy, right? So she tells the story of that. And then my other guest, I can't remember his name, but he's with a biotech company, and they're trying to figure out how to deliver the CAR T receptor to T cells without having to remove the T cells from the patient and infecting them with a virus and then putting them back in. So I don't want to go through the whole thing of what is a CAR T, but for cancer therapy, the T cells often lose track of what they should be killing, and they don't kill the cancer cells. But you can take an antigen from the cancer cell surface and make it into a hybrid molecule called a chimeric antigen receptor, CAR, and then you can put that into, you take T cells out of the patient, you put that gene in and confirm that its protein is being produced, and then you put it back in the patient. Well, it's a little tedious, right? It takes time. And so could you just infect patients with a virus that would go to T cells and put that CAR T cell receptor in? So that's what he's working on. It sounds interesting. So the two of them will be a fun talk. So if you're in Texas, we have to register for South by. It's expensive, and you're probably not going to go. But if you are going, I'm there, and you can find the session on the program. All right. So that's that. Let's see. Is there anything else? Yeah, I wanted to just want to do a mini thingy here. It's only 8:30. We can do this. A mini lecture, like we've done before. Many of you have said you like the mini lectures. So I am going to do a small mini lecture right here now. And I think that's good. That's big enough. So today I gave this whole lecture in my virology course. It's called Infection Basics. How do I change the slide here? I need to have the bloody navigator on the left. Okay. Because I'm not using the usual screen presentation. It doesn't work with this streaming software. So we spent the first part of the course, the first 11 lectures, talking about infection of viruses in cells in culture. So here in this slide, there's a culture dish with cells in it. And you can infect them with viruses and study virus reproduction in them. And now in the second half of the course, we talk about infections of organisms or hosts, mammal hosts mostly, and mostly human hosts. And it's a very different situation, right? Because the cell culture is a dead end. It's a purely experimental situation. You put viruses in, they reproduce, and then they stay there. Unless you take them out and put them onto another cell or in an animal. But there's no natural transmission of the virus from host to host. But in the host, the virus has to get in. It has to find cells that it can reproduce in, and then it needs to get out. So getting out of the host is even more important than getting in, because if you don't get out, you're done. It's the end of the line for you as a virus. And unless there are many other hosts infected, that's the end of the line for that virus. So in this lecture, we talk a little bit about that. And I want to just give you a few slides from that lecture to establish some principles, okay? And I start this course by saying we live and prosper in a cloud of viruses. And live and prosper are key words, because most virus encounters have no consequence. They have no consequence. There's some that are pathogenic that cause disease, of course, but most, the virus comes in us, it reproduces, and you're fine. And so that's why we live and prosper in a cloud of viruses, because viruses are all around us. Now, many viruses infect us and cause no symptoms. They're inapparent or asymptomatic. So let's define what we mean by a symptom, and then there's the other descriptor, the sign. So a symptom is apparent only to the patient. Only you know what your symptoms are. So you have a stomach ache. You have a pain in your leg muscle. You have a headache. Those nobody could know by looking at you or measuring anything. So that's a symptom. And that's going to be important in a moment when we talk about incubation period. Then we have signs of infection. That can be seen by others. And as we get more and more technological, we have assays developed that can get all kinds of signs that we never could before. So what's a sign? Well, let's say measuring virus RNA or DNA in the blood. You can't feel that, but we can measure it. That's a sign of an infection. The production of interferons. I mean, you do feel the fever of interferon, but you don't know that it's actually interferon specifically. So all of those things that you can measure are signs of infection. So someone else can figure it out when they take your blood, for example. They can figure out what's going on in you. So signs and symptoms are very different and very specific for a reason, because an asymptomatic infection means there's no symptoms, but they're clearly signs because you're infected. So the virus is reproducing, the immune system is engaged, and so forth. So let's have an example of an asymptomatic infection. And West Nile virus is a great one because this virus was absent from the U.S. until 1999. In fact, absent from the Americas until 1999, when it was introduced by a traveler, most likely. It's an interesting situation because West Nile virus, we talked about this on a recent TWIV, is transmitted among birds in what we call an enzootic cycle. The mosquitoes bite birds and transfer the virus between them. And periodically, that mosquito will find a human and bite it and deliver virus, and the human develops West Nile fever. But that's the end of the line, because that human usually doesn't have enough viremia for another mosquito to bite it and take enough virus to transmit. So every West Nile infection is a spillover from some bird, probably via a mosquito. So how did West Nile virus get into the U.S. in 1999? Well, first of all, it's identical to, its genome sequence is identical. The virus that came into New York is identical to a virus from a goose on an Israeli goose farm. All right, so how did that virus get here? So it can't be a mosquito. I doubt a mosquito would survive the flight and then fly out the door and go bite someone. Or they could bite someone on a plane. But as far as I know, the first cases, they hadn't just flown in from Israel. But Dixon mentioned on TWIV the other day, young children have higher levels of viremia than adults. So maybe a kid was infected and came over on the plane, and that kid then went out and was bitten by a mosquito somewhere in New York, and then that started the chain of infection. Anyway, in less than 4 years, it spread across the U.S. You can see these maps showing the spread of West Nile from New York initially all the way across the country. And about 1 million people were infected by October of 2004. We know this because we did some antibody surveys, and we can calculate the percent of people who are antibody positive. About 20% of the people developed a febrile illness, and they were also shown to be positive, seropositive. 1% of the people had some kind of CNS disease like paralysis. But, 80% of the people had no symptoms. They were infected, we know, because they had antibodies to West Nile virus. And this is a perfect example because the virus was not here before 1999. Okay? So we can say that they became seropositive by infection with West Nile virus. Now, I know that Amy is thinking, Vincent, how do you know they weren't cross-reactive antibodies to another flavivirus? And absolutely possible. I don't know I'm sure you could probably distinguish that. Nevertheless, the point is that many people have asymptomatic infections, and they have no obvious disease. And for West Nile, it's not how it's spread by asymptomatic infected people. But for SARS-CoV-2, asymptomatically infected people can spread the infection and their problems, their public health problems. So this brings us to the incubation period, which also involves symptoms. The incubation period is defined as the initial period before symptoms of disease are obvious. Symptoms, that means before you feel something. Signs are present. As soon as you're infected, within probably hours, there are signs. The virus is reproducing, your innate immune system is responding. Yet, it's still an incubation period until you get those first symptoms, whatever they may be, a cough or a sneeze or a runny nose. Then you have disease onset. And remember, a lot of people never get that disease onset. They remain asymptomatic. But this is defined by symptoms, not signs. That's an important distinction. The incubation period is the period before symptoms of disease are obvious. And now a key point here is that the virus may or may not be transmitted during the incubation period. It depends on the virus. SARS-CoV-2 is certainly transmitted during the incubation period. And I will show you a slide of that in a moment. And Ebola virus is not transmitted during the incubation period. So it depends on the virus. Let me show you two numbers here that will emphasize that. All right, so here's the relationship between virus shedding and transmissibility. So the incubation period, there are three viruses shown here. The incubation period is shown by this transition from blue to purple. So, I'm sorry, the incubation period is the blue, and the symptom onset is the purple. So the incubation period is blue. So here's SARS, the original SARS 2003. The shedding is shown in the curve, and you can see the shedding is almost non-existent before symptom onset. So there's no pre-symptomatic shedding. And in fact, the peak of shedding occurs at the peak of symptoms, and those patients are pretty sick. They're hospitalized, and we were able to contain that infection quite readily. And that's why it did not spread very much, only 8,000 cases globally. SARS-CoV-2, different story. Here is the infection here, and then we have our incubation period in blue, and then the symptomatic period. But look at the this shedding, which is this curve at the bottom here, begins before the onset of symptoms. And in fact, symptom onset is roughly at the peak of shedding. So you're shedding virus before the point of symptom, the onset of symptoms. And so that's why it's a big problem, because you can be walking around feeling fine and shedding virus and infecting others. We call that community spread. And we did not have community spread with SARS-1 in 2003. And for comparison, here's seasonal influenza, where we have our incubation period and then onset of symptoms. And you see there's a little bit of shedding a day or two before. So not as long as for SARS-CoV-2, but there is still pre-symptomatic shedding for influenza virus. And so people do spread it. They go to work feeling fine, and they're actually shedding virus and infecting others. So that's why incubation period and shedding and transmissibility are important to understand. And one more thing about incubation periods, they vary. And they vary among viruses, as you can see here. And within each virus, they vary as well, right? They can, for Ebola, it's between 2 and 21 days. That could be because the hosts are different, right? And you always look at multiple hosts and you make an average. Could be differences in the virus, but less likely. I think it's a host thing. Or it's also a function of the dose of inoculum, right? How much virus do you take? And so you can see short ones, influenza, rhinovirus, Ebola is not so short at the upper end. But respiratory diseases, they all tend to be short because replication at the primary site is what produces your symptoms. And then the longer ones, usually the virus has to go somewhere else in order to cause the symptoms. And so measles, for example, has to get in the blood and travel to the skin before you get that characteristic rash. Then we go all the way down here to the hepatitis viruses, which have long incubation periods, rabies, 30 to 100 days, and then papillomaviruses, the viruses that cause warts. Many, many days before getting a wart after you have picked it up. And I tell my class, warts, you can have warts anywhere on your body. The ones on the bottom of your feet, you typically pick up by walking somewhere barefooted where there are pieces of human skin. And, we shed skin all the time. That's how the papillomaviruses spread. They make their way to the top layer of living cells. Then those cells die and fall off. Your outer layer of skin is always falling off. It's the dust you see on your furniture, for example. That dust can contain skin cells that have papillomaviruses in them. So in a locker room, for example, people walk around barefoot and they have warts on their feet. They shed skin with papillomaviruses in it, and then you walk by, you have cuts on the bottom of your feet. You pick it up, and you're going to get a wart. So wear foot protection. Wear those flip-flops that you can go in the shower with. Don't walk around barefoot. You're just asking for warts. Asking for warts. All right, so that's what I wanted to talk to you about tonight, and we can actually we can actually continue that discussion next time. But those are really important principles, I think, for you to understand that will make it easier for you to understand terms like incubation period and transmissibility and so forth. All right, let's go to some of your questions. Rach says, on any of your other podcasts, have you ever looked into the hard problem of consciousness? I just watched Dr. Sam Parnia discussing what happens to the brain during the death process. No, I mean, it would be appropriate for This Week in Neuroscience, right? But we haven't talked about consciousness on that at all. You know, if there's a good paper, we will do it. But we have the neuroscience crew has not found that. By the way, on Twin, Vivian is returning, so that'll be fun. So those of you who listen to that know who Vivian is. Bubble tea. We're talking about bubble tea. I'm scrolling through to see what's interesting. What Vanity says, very interesting opinion piece on measles sequelae. Have you heard of James Cherry, UCLA? I have heard of him. I haven't read this piece. But I'll tell you, there are serious sequelae of measles. First of all, the erasure of your immune memory because it destroys memory B cells. And you can get encephalitis in 1 in 1,000 cases. And immunosuppression. So you get susceptibility to other infections that you wouldn't normally have. So there are severe sequelae. You can also get measles pneumonia. And then, of course, the long-term sequelae, which happens many years later, subacute sclerosing panencephalitis, SSPE, which is a fatal degenerative neurological disease. It happens in kids who've had measles and recovered maybe 6 to 10 years afterwards. And they're uniformly fatal. So, you know, we haven't seen a lot of those cases recently, but we're going to, because now we have more cases of measles because people are not vaccinating their kids. And we're going to get SSP, which is horrible disease. I do not understand why you want to convince people not to vaccinate their kids and save their lives. There is nothing caused by vaccines that happens on a population level other than rare side effects like myocarditis in mRNA vaccines for SARS-CoV-2. But this idea that vaccines called this widespread this or that is absurd. It is totally absurd. There's no data supporting it. Why do you have to spread false information? Especially RFK Jr., who says Hep B vaccine caused SIDS, sudden infant death syndrome. No, it didn't. SIDS was caused by kids sleeping with their face down. It had nothing to do with Hep B. In fact, you want to give kids, newborns, Hep B vaccine because they could pick it up from their mother in utero, or they can pick it up from their parents on their saliva if they are infected. It's for them, to protect them. So I don't get this. Anyway, I haven't read that, but I'll take a look. Thank you. All right, Kang has a question. I get confused about how to use the words infectious, contagious, and virulent. Let's try and do that for you. So let me take virulent first. So virulence is the capacity of a virus to cause disease, okay, which is may also be called pathogenic. Pathogenic means a virus can cause disease, and you can quantify virulence in different ways with animal models and so forth, but you cannot quantify virulence in people because you can't do the proper experiments. And if you observe people in a hospital, you you can't rule out confounding effects. You can't say that Omicron is less virulent when it first came out, which is what they did in South Africa. No, it's not possible. So virulence is the capacity of a virus to cause disease. In order to be virulent, the virus reproduces in the cells. Now, an infectious virus is able to enter a cell and reproduce. I would say infectivity is a matter of, in cells and culture, it's a matter of getting in and back out. Many of us have had this discussion. What is infectious? I think it's slightly different in a host, where infectious is the virus can enter the host and then get into a cell. And maybe that's enough for it to be infectious. But in a cell and culture, I think infectivity, because you measure it by a plaque assay, for example, or some other assay, which depends on multiple rounds of reproduction. So infectivity defines the ability of a virus to enter a cell and reproduce. Contagiousness is often talked about in populations, where we say this virus is contagious because it can spread from one person to another. Quantifying contagiousness is very difficult. And there are ways that people do it, none of which I like. But certainly viruses can be contagious, which means they move from one people to another. Some may be more contagious than others, like measles virus apparently can infect one person, can infect many other people, because maybe we actually answered this last time. It was Kang's question. Longer shedding, higher titers of shed. Also, transmission in small droplets that remain suspended in the air for long periods of time. So that's infectious, contagious, and virulent, okay? I hope that helps. Kathleen wants to know, is it possible to prevent shingles if you notice symptoms early and begin treatment with Valtrex? Sister has the symptoms but no visible rash on the tricep of one arm. I don't know. That's a good question. The symptoms, pre-rash symptoms. But the virus has been reproducing for a while. Certainly you can reverse the rash by antivirals, right? You can shorten its period of development. So I would say you might be able to stop the rash by giving the antiviral before the rash forms, yeah. But I don't know for sure. If you do it, let me know. That would be interesting to know. Any thoughts on the tetravalent dengue vaccine and the mRNA CMV vaccine reviewed on Puscast? So I haven't looked at the mRNA CMV vaccine. But the tetravalent dengue vaccine looks good. This is being used in Brazil, I believe, among other places. And the results are an improvement over the Dengvaxia, which had a problem with the type 2 component. So I think we're going to look at this at some future TWIV. I think the paper is in the queue. Yep. Centaurs. Yeah, here's Luther, same thing. How about the Japanese dengue vaccine in Brazil? No risk of antigenic sin. I don't think that's an issue with the dengue vaccine, right? Unless you're talking about the serotype differences. You could call that antigenic sin. So the thing is, if the vaccine gives you good immune responses to all 4 serotypes, then when you get infected, you should make a good immune response to whatever serotype is infecting you and block it from causing severe disease. The problem arises when you don't get a good response to one of the serotypes, and that acts as then when you get infected, it's like your first infection, and then a second one can cause more severe disease. Let me see how that works. So you have the tetravalent, and say the type 2 doesn't work so well, but you do get some antibodies. But it's not enough to block your subsequent infection, in which case you get a memory to the other and it causes more severe disease. So what you need is a solid response to all 4 serotypes to prevent that from happening. So it looks like this is good in that sense. It is preventing this antibody-dependent enhancement of severe disease. CYP3, what a good name. CYP3 enzyme. That's really good. Who else has a question? Here we go. Yeah, not surprising. Unfortunately, Nicola says, when I got my COVID shot, I spoke with the pharmacist who gave it to me, and in 5 minutes of chatting, she said that Omicron is mild, COVID came from a bioweapons lab in China, and so on. No one kind of person is immune to this misinformation, unfortunately. Sean says, the other day eDNA came up. Yeah, on our stream yesterday with Nels, environmental DNA came up. So for those of you who missed it, this is very interesting. So the paper we did, so when you introduce DNA into cells experimentally in the lab, one way to get the DNA in the cell is to apply an electric current. You put the cells in the DNA in a cuvette, and then you apply a current, and that facilitates the DNA getting into the cell, and then it can make its get in the nucleus, be transcribed, and eventually make proteins. So these investigators said, well, what about electric eels? Could they facilitate DNA transfer? So they did an experiment where they put an electric eel in a tank, and then they put a cuvette in the tank with electrodes on it, and then they put in the cuvette some zebrafish larvae, and they mixed them with a plasmid encoding a green fluorescent protein. And then they fed the eel a goldfish, and when eels eat, they send shocks. They're stunning their prey or something. Maybe they're excited to eat. And then they took the zebrafish larvae out, and they indeed had fluorescence in the cells, mostly their external cells. So, in principle, an eel could facilitate electroporation in nature, although that, no, you have to do the experiment. And so, why is this of interest? Because there's a lot of environmental DNA, or eDNA, as Sean mentions here. And what is that? Well, when organisms shed cells into the environment or die, their DNA is there as well. And if it's in the water is full of DNA shed from fish cells that come off the fish, or from excrement from fish that has cells that has DNA in it. And you can actually, I learned this in Montana. I went to a talk by a lady, and one of the people who work in the outdoorsy thing. What is that? National park, but not national park, just local parks service type of thing. And she said they take DNA from water to find out what species of fish actually are living in the water. So they can sequence that DNA, which is called eDNA, and tell what fish are living in the particular stream. So it's everywhere. It's in water. It's in dirt. It's probably on surfaces of things. And there's a possibility that could go from animal to animal, or plant to plant, or whatever, and transfer genetic information. So that's why they did that experiment. Okay, so that's eDNA. I thought that was really, really interesting. Okay, so Costella has whack flu, headaches and body aches. It's not whack flu. It's flu. You get body aches with flu. Did you have it diagnosed? Was it flu A or B? I'm just curious. But you definitely get body aches with influenza, and that's a consequence of inflammation, of cytokines being produced that circulate throughout your body and have effects on different parts of you and different organs. So, yeah. It's not whack. That's funny. Whack flu. So Carol says she had measles as a child. I have antibodies. You have antibodies. That's interesting. I don't know, when were they measured? Would I need the MMR vaccine to protect against mumps and rubella? Um, so you had measles as a child. Okay. And yeah, so you would have antibodies against measles. That's fine. And if you had mumps and rubella as a kid, you would no longer, if you had them before measles, the measles would have erased your immune memory. But if you never had mumps or rubella and were never vaccinated against it, you should probably get vaccinated, right? It's very low chance of infection. Mumps and rubella, not like measles, there's no resurgence despite not immunization. But I think it would be safe if you've never seen those two viruses to get them. Yeah. I mean, it's more of an inconvenience than anything to get these infections. Rubella is a concern mainly in pregnant women, right? That you can have congenital birth defects as a consequence, but obviously that's not a concern in a person who's not pregnant or will not be pregnant anymore. Jessica is reading The Eighth Day of Creation. Yes. Someone sent that to me years ago. It's good. It's a good description of the early days of molecular biology. Yeah. It's good. Gerbiest. I'm a young lecturer at a university in Canada for virology, and your lectures have been a guide for my first time teaching the course. Thank you. Oh, you're welcome. I'm glad they're useful. That's one of the things I like that people do with the lectures, is to use them to teach their virology course, because not everyone is a virologist, right? And so I can impart what I know to you through the lectures on YouTube. And many people call me and say, can I use them? Blah, blah, blah. Yeah, of course you can use them. I'd like you to give credit, but even if you don't, you can still use them. You don't have to pay for it or anything. It's my gift to humanity, right? This stuff, what did I call it? Wandering around in my head. Some of it I put out, and I wonder what fraction gets out in my lectures and in my podcast and in this live stream. But you're very welcome to use it. Yeah. Paso Robles. Okay, Jonathan, if some viruses uncode at low pH, could they be tricked into uncoding by bacterial acid production locally in the mucosa? So fusion is highly regulated, and you typically need something else besides the low pH to catalyze fusion, right? And so just exposing viruses, you need, say, a receptor interaction. So receptor, if a virus is bound to a receptor and then you lower the pH, that will lead to fusion. And in some cases, you need cleavage as well. Or in some cases, you need a second protein to allow the fusion to happen. So the virus binds the receptor, and then a second protein interacts, which exposes the fusion peptide. So it's regulated because you don't want viruses bumping into any membrane and fusing with them, because that would be non-productive and a waste of genetic energy. So it's regulated. So just lowering the pH won't do that unless the viruses are bound to cells. And, you could do it in a mucosal surface, but the receptor for the virus has to be there and engaging the virus for that to work. Costello said, dinner is done, ready to sit and chill and listen. Very good. Enjoy your dinner. So here, since I've been transmitting at the incubator lately, I get dinner beforehand. So I usually come back from my class around 6, and I get dinner. There's a restaurant down below. I'm very lazy. It's just right here next to the door of the incubator. And so it's called Shanghai what? I don't even spice or something, and it's good. So I go there. Let's see. The slides are done already. I'm sorry if you couldn't see them. They're already pretty big. Um, it's this transmitting program is not great for showing stuff. I'm sorry that they were small. And I just saw your message now, so sorry about that. So Rach says, I wondered if you had heard of Dr. Sam Parnia and his work on the dying brain. All right, now I lost your comment. Where is it? Sorry, I made the font bigger and it went away. Oh, there we go. Looking into where consciousness is found in the brain. Yeah, it's a very hard problem. Where is it? I don't know Sam Parnia, but I'm going to write his name down and check it out for Twin and put it right next to the virtual incubator tour, Sam Parnia, for people who aren't going to visit or have no opportunity to. But if you're visiting New York, you should make it a destination. It's a good place to visit. Okay. Hans says the mere presence of a virus in a tissue is insufficient proof of it being infectious. That's correct. What is necessary for a virus to be infectious? Are all virions infectious? Well, those are good questions. Are all virions infectious? No, they're not usually, and it depends on the virus. So some are worse than others. We actually measure this. We actually measure this by what we call the particle to PFU ratio. So if you have a tube of virus, okay, so here's a glass of virus, it's a lot of virus, you can count the particles. It's very hard to count the particles. So you usually just do this once for any particular virus. You usually could put them on an EM grid and count them, right? Or you could do spectrophotometric methods. Anyway, you count the number of particles, and then you measure their infectivity. And if every particle is infectious, the number of particles it will equal the number of infectious units, right? There are very few viruses that are like that. There are some. Some leaky forest virus, about half the particles are infectious. It's 1 to 2. And for a bacteriophage Phe6, the particle to PFU ratio is 1, so every particle is infectious. But for other viruses, you have hundreds of non-infectious particles and only one infectious particle. And why that is a bit of a puzzle. There are errors made during assembly, there are mutations in the genome, particles break during transport and so forth. So a lot of reasons why particles would not be infectious. So it really depends on the virus. If I remember polio, 1 in 300 are infectious. And for other viruses, 1 in 10,000 are infectious. So a lot of material is wasted, but that seems to be an error-prone process. So what is necessary for a virus to be infectious? You need the full genome, whatever that may be, in the particle, without anything missing, without the ends missing, without nothing in between missing, without anything in between missing, without any mutations that would inactivate infectivity. It has to be the right genome sequence. Then it has to be packaged in a particle. And the particle packaging has to be correct structurally. The proteins have to be right, form a good shell that's stable. And if you have a membrane, that has to be correct with spikes in it as well. So basically, you have to make what looks like a mature particle. And then the thing is that many of the non-infectious particles look perfectly good. You can't tell what's wrong with them. But you need the genome and you need the shell around it. And that is what you need to have infectivity. This is a good question. They're all good questions. But that was a teaching moment. So Razor says, would it be possible to detect in the population persons that have not been infected with SARS-CoV-2? If so, if it is, would these persons be scientifically interesting to study, whether vaccinated or not? Well, up until recently, Daniel Griffin said he didn't have SARS-CoV-2 infection, right? And Kathy Spindler has not been infected. So there's certainly people out there. Now, whether they actually have not been infected or not is a good question, right? Because they may have been infected, but they just didn't know it. Maybe they had an asymptomatic infection. So you could look for antibodies against nuclear capsid protein because they have certainly been vaccinated, right? So you can't look for antibodies against spike. But you could, and Kathy or Daniel, before Daniel was infected, you could say, oh, you don't have antibodies to nuclear capsid protein, which, by the way, is absent in the vaccines, most of the 2 mRNA vaccines anyway. Then you certainly haven't been infected. And then what would you do with them? There have been parts of studies of vaccines. I saw a talk today where the person had 3 groups of people they were studying. They had people who had been vaccinated 2 or 3 times, and then people who had been vaccinated 2 times and then infected, and then people who were just infected, and comparing antibody levels in them. So they can, if they haven't been infected but vaccinated a few times, they can be part of that study to say, what kind of antibody response do you make if you've been vaccinated but never infected? So, I mean, interestingly, in her data, the amount of binding antibodies to spike were always higher in people who had been infected. And even after 2 vaccine doses, it was never quite as high. But the 3rd vaccine dose pushed it to equality. So now they all have the same level of binding antibodies. So that, I think that's where those individuals would be useful for study. But they are not too many and hard to identify. I don't know how you would do it. You'd have to do it probably in a healthcare setting, right? But it was interesting from this talk that New York is the biggest mixing center of people in the world over every other city. More people come here from different countries than any other city in the world. So we get viruses from everywhere here in New York. And this person found, the first case of COVID in New York, I think, was in March. And they, by screening antibodies of people who went to the hospital, they found antibodies to SARS-CoV-2 back in February. Not surprising, right? The first official case in New York City was in March, but there were people infected before. They just had asymptomatic infections. But then when you check their blood for antibodies, you see them. So New York is, I didn't think it was. I thought some other city would be, but New York is the big one. And everything comes here, and then we spread it to other places as well. So it would be nice to do more screening of people coming into the city to see what's happening here. Okie dokie. There are plenty of questions here. John says, for a normal vaccinated person encountering measles, will they clear the infection with no symptoms typically? Or will they feel ill, and does it take 2 weeks for the adaptive immune system to clear? I think they will clear it most likely with no symptoms, or maybe flu-like symptoms, the earliest symptoms of a virus infection, fever, malaise, muscle aches, where you can't distinguish what virus is doing it. But you certainly won't get the rash, and you won't get the nasty sequelae. But it could take a week or two for all that to clear out, but you're going to be feeling pretty well. Yeah. Podcastologist. There it is. Stuff spinning around in my head. That's it. It is spinning around. It bumps into each other. Sometimes they recombine, so I get confused and I say things. Oh, look at this. The virus-infected cells use more nutrients to replicate virus. This is like last Monday's lecture. The infected cell. Absolutely. To make a lot of virus particles, the cell's metabolism has to be cranked up. Has to raise the thermostat for sure. And so, yeah, they have to make amino acids, they have to make lipids, and they have to make nucleotides to make nucleic acids, right? And lots of other molecules as well, but those are the big ones. So, and they need energy to make virus particles, ATP, NADPH. So, yeah, many viruses stimulate glucose uptake into the cell, which goes through the glycolytic pathway and ends up with lactic acid at the end. But in the process, it's aerobic respiration. Is it aerobic? Forget it. It's just glycolysis. And you get lactic acid in the end. And you release ATP and NADPH along the way, and you make energy. And that pathway, actually, you can shunt off and make the pentose phosphate shunt comes off glycolytic pathway, and you can make nucleotides there with that. And then you could take, instead of going all the way to lactic acid, you can take pyruvate and convert it to precursors of lipids. So that one pathway is very powerful. Then, of course, you have the Krebs cycle, which takes as an input acetyl-CoA from the pyruvate from the from the glycolytic pathway, and it converts it into various compounds and makes energy as well. And many of those can be shunted off to make lipids and so forth. So totally using more nutrients for sure. A virus-infected cell, its metabolism is cranked up. And you can see that in when you have cells in culture, we use a liquid medium that's red. It has an indicator, a pH indicator in it. And as a virus-infected cell is incubating, they turn yellow because of the lactic acid being produced. Yeah, you have to give them a lot of food in order to make viruses. Yep. What's the science behind the CDC's most recent recommendation that older adults receive another COVID booster? So the science is minimal, but it's observational in that as you are older, you have your duration of immunity is lower. So your immunity to previous vaccination is lower. But more importantly, another booster with the Omicron variant is what you're talking about, I presume, because if you had the original two, you should always get a third because that really diversifies your antibody response. But now we're talking about the Omicron-specific booster because Omicron is so different that it evades immunity, antibody immunity, and you get infected. More people get infected. Now, you don't necessarily get more disease unless you are older. So people who are older have weakened immune systems. They may have comorbidities. And so even a rather mild infection, as Paul Offit says, can send them in the hospital because they just can't tolerate any kind of infection, which a 40-year-old would handle no problem. So that's why the recommendation exists, to boost their immunity. And, you know, the thing is that the virus is changing continuously, so there will be more variants probably in a year that evade that immunity as well. So we'll have to see if we have to keep updating this vaccine every year. But that's the idea, is that people who are older don't do well with the simplest of infections. So give them some immunity, yeah. Mm-hmm. Climatology is another one. All these words want to make me learn Latin. Yeah, that's cool. Yeah, it's very cool. Okie dokie. Oh, there you go. Calliope, poliomyelitis, from the Greek polios, gray, and muelos, marrow. The itis is inflammation. I think the itis is Latin, no? Marrow, polios is gray, and muelos is marrow. They're both Greek. Okay, maybe the itis is Latin. Thank you very much. That's exactly it. Yep. Got it. Have you ever come to Cincinnati? I don't know if you're talking to me or Nicola, but I have not been to Cincinnati. But I'm going to Columbus. It's got to be close to you, right? Why don't you come see TWIV in Columbus, Ohio this summer? Uh, let me look it up. What month is it? I think it must be ASV Columbus is June 24th to 28th. Why don't you come to that? I'll get you in. Because Lise is coming, and I think there's other people. Well, Steph Langle's coming from Cleveland, and there's another person here coming who's at the University of Toledo. So I don't think that's the name of it. It's the University of Ohio in Toledo. I don't remember, but you're here. Artemis is ichthyology. I think we missed that. The study of fossil footprints, tracks, and burrows. Ichnology. Oh, so that reminds me. I was on a plane, and you know those red bugs that are plaguing the Northeast, lantern bugs, right? There was we were sitting on the runway, and there was one on the window, and this young guy sitting there. He couldn't kill it, which I don't blame him. I wouldn't kill it either. So the flight attendant said, "Is there an entomologist in the house?" And the guy got up actually and went over and grabbed it. He wrapped it in a piece of paper, and his partner said, "It's a good thing you didn't ask for an ichthyologist." And the flight attendant said, "What's that?" You know what an ichthyologist is? A study of fishes, right? Here's a question that I don't know the answer of. Does the study of the different mass extinctions all have their own names? We got fossils from the Ordovician extinction here for days. I don't know. I would guess they did, but someone might know here. My favorite new profession is netnographer, an anthropologist who studies what people do on the internet. Cool. Oh, Troy, you coming to TWIV and TWIVO? Very good. Looking forward to meet you. 714 was my all-time favorite TWIV. TWIV 714. Oh, yeah, that's The Shape of Spike with Jason McClellan. Yep. So that Jason will be back. Yep. It was a nurse that didn't know what a virologist is called. Campbell. No, it wasn't John Campbell. No. But maybe John might know by now, but who knows? Yeah, we make designer good viruses. Lots of people are working on that. Yeah, for sure. Yep. Paige likes the lecture. Thank you very much. It was a little short thing because I want to answer questions here, but we'll do more next week on the same topic because it's nice to take little chunks, right? Everybody like that. Good. Excellent. When my 6-year-old was out of school for a week with this whack flu. Okay, you gave it a name. I kept playing the first 3-4 lectures for him. That's cool. Okay, this is a good question from Artemis. So fever, when I don't feel feverish, is a sign. Yeah, because you have to measure fever. But some people can tell they have a fever, right? So it's one of those gray areas. Some people can tell they have a fever, and you could tell the doc, I feel feverish. Well, they would measure it then, and it would be a sign. Yeah. I love this quote, right? We live and prosper in a cloud of viruses. We do. Even the worst viruses do not kill most people, right? The worst was maybe 1918 in terms of viruses. I mean, COVID was horrible, but it just kills a fraction of the population, and viruses are not going to kill everyone. They're just not doing it. Here we go. Ioto 2001, the crows are returning here in the Midwest finally after being decimated by a West Nile virus. Many crows and many other North American birds were killed because they'd never seen the virus before. But birds that had been raised in other countries like Africa and the Middle East, where there was West Nile virus all the time, and those birds at the zoo, they survived, and the others, North American birds, did not. Man, I was sitting outside Monday when it was like 16C and kept getting mosquitoes biting me already. I was so sad, like it's February. Go back to hibernating or whatever it is mosquitoes do. Yeah, they basically go and hibernate, usually under the leaves in the woods, and they go into a suspended state. Dixon was talking about this on the last TWIV. They also go in the subways here in New York and just stick on the walls, and they live and they don't need many nutrients and so forth. And then when it warms up, they go back. Yeah. Sorry, the slides were too small for you, but you could hear me talking. Could something in the innate immune system be used as a flag for some kind of infection? For sure, yeah. You could, if you could rapidly measure interferons, you could, but usually we know otherwise, but not have to do that. But yeah, it could be done. How do you distinguish prodrome from incubation period? So the prodrome is the period of symptoms. So you're out of the incubation period now. You're in the symptomatic period. It's a part of the symptomatic period where the symptoms are non-specific. You can't tell it's measles or smallpox or whatever, like flu-like symptoms, fever, malaise, muscle aches, and so forth. And then when it becomes characteristic, it's no longer the prodrome. Then it's like measles and polio and so forth. Because before you get paralyzed, there's a flu-like symptom phase as well. If someone is infected but does not develop disease, would you say that they only had an incubation period? No, because the incubation period is defined in the context of symptoms, right? So if you never had symptoms, then there's no incubation period, right? There's no, and you may have been infected, but since you never develop symptoms, by definition, there's no incubation period, because that's just the way it's defined, right? It's kind of a funny way of doing it, but virus is certainly reproducing in you, and eventually it's eliminated, but we don't call it that, yeah. What are the details on the March 11th event? Here you go, Garrett. Let's bring it up. March 11th, that's Monday, at 11 a.m. in Norman Hackerman Building. So that's a building you will find on the campus of the University of Texas in Austin. And then there's a big seminar room there, and you can't miss it. Okay, it's probably close to the entrance. So there it is. All right. My background looks amazing. Thank you. It's always the same stuff, but I change the colors just to lighten it up, to spice it up. I have a couple of tube lamps on the shelves, and then there's a light hanging, not the white one there, but another beyond that, splashing light on the wall. So thank you. I'm glad you like it. Can we talk about eDNA? We did, right? I hope you heard it. We talked about it quite a bit. Must the skin be cracked to acquire the wart? Let's take that first. So the outer layer of skin is dead, yes. So if it's not breached, the virus won't get across it. But of course, needle sticks get virus across it, animal bites, mosquito bites. But if it's not cracked, the virus will not get into the underlying live cells and can't replicate. So it has to be cracked. But you have micro cracks on your skin that you can't even see, right? If you have particularly dry skin, you can have a lot of cracks in the outer layer, which is dried out and cracks, especially in the bottom of your feet. I mean, our feet can be a nightmare, right? If you don't take care of them, they get lots of callus and it cracks. And some people I've seen, their heels are just cracked right through to the underlying epidermis. Oh my God, because the callus is so thick and you see the red flesh underneath it. So, well, I don't mean to gross you out, but that's not what my feet look like, by the way. My feet are beautiful. I take care of them. And because I used to rip my sheets. I had such calluses, I ripped my sheets just sleeping. So I said, this is ridiculous because, sheets are expensive. And I just take care of them, and now they're very smooth and no more sheet ripping. It's probably TMI. I'm sorry. But yes, the skin must be cracked. What can you get from sitting on a toilet seat? The toilet seat itself is not really the risk unless it's filthy, right? If someone has defecated on the seat, which I see all the time, you go in and people don't clean the seat off. Come on, people. You should clean it off. If you've got wipes with you, that would be great. Because, you get fecal material from another person, and they may have some infection that you don't want to acquire by sitting on. All right, now, the water is the issue. Because when you sit on the toilet and you defecate or urinate, that makes an aerosol, and it can generate an aerosol with whatever viruses are in the water. So even if someone has flushed the toilet, they're going to be viruses stuck to the porcelain, and they're going to still remain in the water for a long time. So you can get those. And so what can you do about that? I don't think there's anything you can do, frankly. I know some people tell me they hover, right? They never sit down, they hover. So you get this vision of someone, with their butt up in the air. I'm not sure how effective that is. I don't know how far the aerosol goes, but I don't know what you do. I mean, even in a urinal, there's some kind of air, standing up, there's some kind of aerosolization. So basically, public restrooms are pretty iffy, and they're filthy because they're not cleaned very often. They are cleaned often, but not enough. They'd have to be cleaned every hour or two. So the surfaces are contaminated. The water in the toilets are contaminated. So, on the other hand, you should not hold it for long periods of time because that can cause other issues like urinary tract infections. So, the splashing of the toilet, I don't know what to do about it because you need it there. So, but you can get, I know stories of people getting urinary tract infections from using a toilet in a hospital room of a patient who had a UTI. I remember hearing this at a talk years ago by a physician scientist who studied UTIs, and he said this. So in women are more likely to get UTIs because the urethra is very short and the bacteria can get in easily because urination will flush out the bacteria, but you have to urinate frequently because you have a short urethra. Men have a longer urethra, right? But as you get older, men also acquire urinary tract infections because their defenses are crappy. Yeah, that's the right word, right? So this case was an older man with a UTI had gone to his kidneys. That's really bad when it goes from the urethra into the bladder and up to the kidneys. And he was hospitalized. His daughter came to visit him. She used the toilet and got his E. coli infection just by sitting on the toilet, not from the seat, from the aerosolization of the water. So I don't know what to tell you. Just be careful. It's enough. Mudslinger. Could ME simply be a non-paralytic form of polio? Myalgic encephalomyelitis? I don't think so. I don't think so. I think, many people feel that ME/CFS, if that's what you're talking about, happens after enterovirus infections, but it may happen after many different virus infections. So I don't know any evidence that it's a non-paralytic form of polio. There are non-paralytic forms, but they're acute infections. They don't persist. David asks, in a prebiotic world, could you have mixed DNA, RNA polymers with variant sugars? I don't see why not. I think what we consider to be nucleic acids back then in this prebiotic world were very different from what we have today. And people study that. So I'm not familiar with the work, but I'm pretty sure they have variant chemical structures. And the interesting thing about them is that they self-polymerize in the absence of an enzyme. So, the enzymes today are needed to add nucleotides to growing nucleic acids. But back then, they self-polymerized. And that can be reproduced in the lab today. So that's quite interesting. Callie says, I will be visiting a relative who is taking immunosuppressants. What can I do to protect them during my visit? We have already gotten flu and COVID shots this past fall. Well, so the person who is immunosuppressed should be fully vaccinated as well. And, they don't respond well to the usual doses that we non-immunosuppressed people get, but extra doses are good for them. You may ask that everyone mask if you're really worried about it, depending on the extent of immunosuppression. There are different kinds of immunosuppression with different risk factors. And we did a paper on TWIV recently where they looked at people with different risk factors, and they have different risks for severe COVID. So I think if you're going to be inside, then, a combination of vaccination and masking is probably what you can do. Will, I stopped listening to your virus course because I didn't understand a word. But your first lecture was brilliant and should be heard by every health journalist. I'm sorry you didn't understand a word. It makes me think I should do a course in really basic virology. I've been talking about this for a long time. Basic virology. I don't know. Someone suggested basic virology, basic virology for regular people. I don't want to disparage anyone because we're all regular. But really, what was the title I came up with? I forgot. Basic virology for something. But I'd like to do that to see if I can talk it at a level that most people can understand. Because this gives me pain, Will, for you to say that you didn't understand a word. It really pains me. I feel I've failed. And I want to fix that. If there were only a way to prevent measles, well, there is a way, and that's to vaccinate. And people choose not to. So go figure. I don't get it. We need the biology course before the virology course. Yeah, I know. I should try and provide the biology you need. I could do that. Yeah. Troy was immunized with MMR 54 years ago. So you probably just got one. And the recent evidence indicates that you need two. And so I would say you should just talk to your physician, because I am not a physician and I shall not dispense medical advice. And some people will say, but Vincent, you have. And I said, no, I haven't dispensed medical advice. I have said it is recommended that you get a booster. Talk to your physician, okay? And the science is that there are outbreaks of measles in kids who are just vaccinated once. And so 2 is now considered to be needed for complete protection against severe disease. RFK Jr. is a virus vector, perhaps more dangerous than mosquitoes. I think he's quite dangerous, frankly. Just says everything that's wrong and dangerous. Tryon says, in an virus-affected cell, is there turnover of copper or zinc? I don't think so. Amy, let me know if you know the answer to that. I haven't heard from Amy here. I know she's here. Let's see if she has answered some of my previous questions. Not yet. Maybe later on in the stream. Virology for everyone. That's a cool title. Jason just suggested that. Kyle says, on the recent SARS-2 infection, line 1 media. Oh, this is the Yanish reverse transcription of N protein fragments. I think this is highly unlikely to occur in actual people. So his system was a highly artificial laboratory system where cells were overexpressing this line 1 element, which has reverse transcriptase. And then you put in RNA and it gets copied to DNA. I think it's highly unlikely that this would end up being integrated. But if it did, what would be the consequence, right? I mean, in an epithelial cell that turns over, little consequence. You could argue it could integrate next to an oncogene, but there's no evidence for that. And I don't, people are looking to see if there are any long-term sequelae. So all I can say is, the risk of infection outweighed the theoretical risk of reverse transcription and integration by far. So that's the way you have to look at everything, right? Everything is a balance of risk versus benefit. And most of the time, the vaccines give you the benefit way over the risk of infection. RFK Jr.'s propaganda comes from a case where a vaccine was reconstituted with a muscle relaxant rather than saline. He was told about the error in reporting, but keeps going. He's an agent of chaos. Yeah. So he then went to Samoa and convinced the government that the vaccine was bad. The MMR vaccine was bad. The immunization rates dropped to 50%. They had an outbreak of measles and 80 kids died because he told them not to vaccinate. And he said, those kids died from the measles vaccine. Well, wait, you told them not to vaccinate. How can it be caused by the measles vaccine? It's just ignorant. And I don't know if it's willful or pure ignorance of not knowing something. I tend to doubt it. I mean, he said that vaccines are never placebo tested. This is just blatantly false information. This is BS. So how can you say this? And people are telling him, RFK Jr., that's wrong. I don't know why he's doing this. It's very scary because a lot of people believe him, right? I was wondering about the shedding of SARS-CoV-2. I only got 3 days off work. I don't believe we have a good measure of shedding and its relation to transmission. But yeah, you can err on the safe side and stay 5 days. Most shedding is over by 5 days. You still see infectious virus past 5 days, but I don't think it's enough to transmit. But again, it would be nice to have those studies, but we don't. Vincent, please elaborate on measles erases immune memory. Memory of what? Of previous vaccinations and infections. So when you are infected or vaccinated, your body makes immune responses to the pathogen, and it keeps a memory of them. It keeps B cells and T cells that have a memory of that pathogen. And if you get reinfected, they can respond really quickly within days, as opposed to waiting weeks to build those new antibodies and T cells. Those cells are wiped out by measles virus infection. So any virus vaccine you've had, any vaccine you've had, any infection you've had, you no longer have immunity. You have to get revaccinated. And, if there's no vaccine, it's risk. So that's why it's bad. Is anyone monitoring all the war zones for emerging virus mutants? Well, I think the monitoring is poor, right? Because it's a war zone. I know early in the Ukraine conflict, there was a case of polio there. You can imagine that vaccination rates for many vaccines are dropping. And maybe we'll see outbreaks. But I don't know that the monitoring is very good because it's a conflict area, right? FYI, some of you may not have seen Paul Offit, an hour-long podcast a week ago. Michael Shermer, skeptic. I think he normally debunks UFO stuff, but he has a big audience. It was really good. Good. That's very good. I've heard that some COVID patients are super spreaders. Well, that's actually a slide that I didn't show you today. So let me just bring it up, and I'll try and make it larger. Where's my super spreader slide here? That's towards the end, right? Where is it? Transmission. Why can't I find a lousy super spreader slide? And I mean lousy. Where is it? For gosh sakes. You know, one of these things where you can't find something and it's right in front of you. Hold on, folks. Hold on. I'm going to go through every slide. Sorry about this. And we're going to find this super spreader slide because I just showed it today. Right? Oh, I see. It's not here because I took it out of the we had a special version of my lecture tonight. So let me open the class lecture and we can look at a super spreader. Yeah, okay, that's it. Here we go. Super spreader right here. So let's see if I can get this to work now and look better. Make it, see if I play, then you can't see anything. No, that's not right. So you'll have to just deal with this. I think it's good enough. Super spreader events, clustering and super spreading potential of SARS-CoV-2 infections. This was a study out of Hong Kong early in the pandemic. Basically, in certain situations, one individual will infect many. So here's one individual, B, in a bar who infected, probably 15 to 20 other people, and they went on and infected others. They did contact tracing to sort out all of this stuff. And that person probably is, well, not necessarily. It could be the one infected person, and it's just a crowded situation that leads to super spread. That's why they're called super spreader events, because we don't know that there's anything special about those people. You may say, oh, well, they shed a lot of virus, but not necessarily. Okay, so that's super spreader stuff, which basically is you have bars, temples, weddings, families, work, etc. Those are the situations where you get super spreader events. You introduce one person into that, and they have a contact with a lot of people, so they have a super spreader event. I didn't know that about B-cell immunity and measles. To the anti-vaxxers trying to strengthen their immune system, this would be good for them to know. They don't believe us because they think we're being paid by the vaccine manufacturers to say that, okay? Did you get my email suggesting an episode to discuss a Nature article about senolytic CAR T cells to reverse aging? It didn't. I'll go look for it again. I might have missed it. Yeah. I'm not going to answer that one. It's probably been removed anyway. Yeah, CDC director said today another dose of the updated vaccine is available over 65. Won't imprinting occur? I don't think so. It's now you've already had one dose of this. You're going to get another. Just going to get a memory response. So I think it's fine. Can I explain what a memory cell is? All right. So when you're infected with a pathogen, you have white blood cells of two different kinds that respond. One of them are B cells that make antibodies, and then the other is T cells, which can also have antiviral effects, although they don't make antibodies. And those cells work, but they have a limited lifespan. But a version of them is made called a memory cell that lasts a long time. It has longevity, and it remembers the antibody and the T cell functions against that particular virus, right? So whereas initially when you're infected, it takes a week or so, 10 days to make those antibodies and T cells, B cells and T cells. Now if you have them as memory, then they can respond within days. So it's a faster response, and you more rapidly bring down the infection and prevent severe disease. So the memory simply means a memory of the pathogen. For a B cell, it's the antibody that combines with that pathogen, because the B cells have to go through a process of recognizing the pathogen and making the right antibody, and that takes time, making a good antibody. And so you want to store that in a memory cell that lasts a long time. It's the same for T cells. They recognize a certain part of the pathogen, and that memory is stored in them. Jason says, see Michael Levin at Tufts with bioelectricity, pattern memory, cellular communication, excellent questions and presentations. Be a good TWIV conversation. All right, Michael Levin. Got it. Writing it down. How many thought PFU emanating from my brain? I don't know. There's no assay for that, right? SIP3 says, your lectures have changed how I see the world. I can't see. Gives me a better perspective on how life happens. Thank you. That's very kind. I appreciate it. Yeah, weird flu and body aches. Yeah, I've seen that here without respiratory symptoms. But, yeah, it exists. Electric eel creates new species through electroporation. Yeah. Yeah. David Feigenbaum from TWIV 766 was just featured in the Wall Street Journal. I think it's cool when guests show up in the news, and his story stuck with me. Can you bring on Ian? Where did that go? Sometimes when I click on Ian Copeland, PhD, as a guest, who is Ian Copeland? I'll look it up. I'll write that down too. Ian Copeland. I don't know what the incubator is. It's just the name of our studio here, because an incubator is a piece of equipment in the lab where you incubate reactions. And we thought that was a good name for a science studio, recording studio. There you go. If you're here, visit it. Lisa's been here, and so has Tona. And many others as well. I'm in the incubator right now. That's right. It's a cool name. Thanks to Amy for that name. Raphael says, thing I don't like with the dengue vaccine is that we don't have only dengue. The mosquito spreads other things. Yeah, of course. Brazil was dengue-free in the 50s because of DDT. And then we stopped using DDT, and mosquitoes came back. But, yeah, we need other vaccines as well. Yeah, for sure. So it's a matter of QC, quality control and assembling a virus. That's part of it, yeah. But you could look at that as the whole thing. Yeah, it's QC. Can you comment on the IgG4 antibody response to mRNA vaccines for SARS-CoV-2? So some time ago, a paper was published showing that the antibodies you make to spike, which is produced from the mRNA vaccines, is skewed to a certain type of IgG called IgG4, which has different properties from IgG1 and 2. But there's no indication that this is a problem because the vaccine has prevented a lot of deaths, right? It works. So I say it's not concerning until we find out that it is. And there's no indication that it would be concerning. So I wouldn't worry about it at all. No. You have not had COVID to your knowledge. Are there any home tests to check if someone has antibodies? I believe there are home antibody tests, yeah. And it was another question about erasure of immune memory by measles, and I believe I have explained that. By the way, if you want to help us make these programs, we're a nonprofit, Microbe TV, and your donations are U.S. federal tax deductible. You can go to Microbe TV on Venmo, or you can go to microbe.tv/contribute. You can see the addresses right above me here. You can also donate here on the stream with the donate button. Either way, we really appreciate it. And John usually every week donates to Venmo. It's not the $1 billion that Einstein School of Medicine just got, but we appreciate everything. And all of this studio you see here was made from your donations. Can the measles vaccinated years ago spread to non-vaccinated people? I don't know what you mean by that. if you get vaccinated, can you spread measles? No, I believe that doesn't happen. I asked that of Paul Offit, actually, and he said it doesn't happen. Abdul Aziz, thank you for your support of science communication. Do a podcast with immunologist Andrea Love. Okay, I'm writing the name down and I will check it out. But thank you for your contribution. Really appreciate it. Here we go. Aerobic respiration uses oxygen as the terminal electron, except the pyruvate accept electrons to give lactic acid. So it's not aerobic. Thank you very much. It's the pyruvate that accepts the electrons and you get lactic acid. Yep. Did all the countries that chose not to give the new COVID vaccine to kids get it wrong? Any COVID vaccine. If you don't give any COVID vaccine to kids, you got it wrong. But if they've gotten 3 other doses and not this one, I have less of an issue with that. But no vaccine at all is downright ignorant because kids die of COVID. And if it's your kid, you're going to feel really bad about it, aren't you? Thank you, Kip, for your contribution to science communication. Really appreciate it. Is lactic acid the reason for achy muscles? I don't think so, because most of the viruses are not replicating in achy muscles. I believe that is a consequence of inflammation and cytokines and chemokines, mostly cytokines. They're diffusing throughout your body, and they are causing the muscle aches. Yeah. Thank you, Catherine, for your contribution. We already answered your question. Sometimes it just takes me a while to get through the queue, but we appreciate your contribution to science communication. Heard in a dental lecture, we make 100 to 120 pounds of ATP daily. Make a lot of ATP. We make a lot of neutrophils, too. Listen to the latest Immune. You make 1 billion per kilogram of your body weight per day from the bone marrow. Okay. Artemis says, knowing Greek helped me with my micro exam. When the answer to a question about fungal spores was arthroblastoconidia, I don't remember the exact question. All right. Leo, thank you for your contribution to science communication. Appreciate it. Unfortunately, anti-vaccinators are coming to ASM in June via the abstracts. I will try and find the author and email you later. Wow. Please do. Vincent at microbe.tv. Tour of the museum shelf will be fun. Yeah, I'll make that a part of the virtual tour. I'll tour everything. Callus, not fecal material. Callus on my heels ripping the sheets. You think the worst of me. I wouldn't say that. Would it do anything to carry a spray bottle of bleach? No. You don't need that. The water you're not going to help with the water because then you're going to aerosol bleach onto your delicates there, and that's not going to be good. So I wouldn't do that. Do hand dryers in restrooms potentially disperse? Yeah, they do. Those studies have been done, and they really disperse stuff. It's really better to have, unfortunately, paper towels for that, but, that's environmentally unfair. The aerosols created from the toilet should be contained. The oral cleansing tools should be kept in direct sunlight. In a different room is best. That's what Michael Schmidt does. And, by the way, closing the toilet doesn't matter. A study we just did on TWIV. Story we just did on TWIV shows that when you close the lid, as much comes out as when it's open. So, she should have hovered. I don't know if hovering matters. I guess a lot of people hover, right? The great George Carlin once said he never washed his hands after leaving a public restroom because he figured his privates were the cleanest thing. Oh, my God. Oh, my God. Okay. Basic virology. People are signing up for. I will do it. I don't know when. I will try and do that for you. Virology for everyone. I kind of like that. Virology for the layman. I think I could do that. All right. I think we will wrap up now. And let me just see. I know there's some great questions left. Margot, thank you for your support of science communication. Character on the floor laughing really hard, probably to one of the things I said, like hovering over the toilet. All right. I want to thank our moderators for tonight. We had with us Steph, we had Andrew, we had Les, we had Vanity Nutrition, and we had Vanity Nutrition, Andrew, Steph, Tom from Wisconsin, who has since left. Thank you all for moderating. Thank you all for coming. Over 200 of you. If you haven't hit the like button, please do it. It helps attract other people to this. Virology for the rest of us. Why is it so difficult to find a cure for HIV/AIDS? We'll see. We'll talk about that next time. Come back with your questions. I'll be back next week. Sounds like a comedian, right? Let me get the calendar here. Just a minute. Now that I'm trying to get the calendar. We will be back next week. What is the date? It is Wednesday. We're into March. March 6th, 8 p.m. Eastern. We'll do another office hours, and I'll try and do a virtual tour and answer some of these questions that I haven't got to yet. Thanks again to all the moderators. Thank you all for coming tonight. Really appreciate it. See you next week. Let me take Margaret off down there. Okay, sorry about that. See you next week. Until then, be safe. Good night, folks.